Good afternoon, viewers. Welcome to MouShauk. And we will discuss today is the topic is the useful role in medicine of the bacteria chapter. We were discussing economic importance of bacteria. So part two, we will discuss that is, it will contain the roles, useful and the harmful roles of bacteria. Okay. So for that, first we will start with the useful roles and then we will come to the harmful effects. Okay. So before that, I would request you to please do subscribe my channel if you are not subscribed yet. And if you are a new viewer to my channel, because that will notify you as soon as a new video is uploaded. And if you are liking the video, press the like button, share with your friends so that they can also be benefited from these videos. And do meditate before you start your day. So for meditation, we will concentrate on a point of light. Okay, think that light is falling on you. Your whole body is being enlightened, feeling happy and joyful. You're getting interest in whatever work you are doing. You're feeling happy. You're getting interest. And becoming successful at the end of the day. You're thanking God for being with you the whole day and supporting you and helping you. Thank you. So for today's topic, we'll start with the useful role in medicine first. So in the field of medicine, bacteria are used to produce antibiotics, serums, and vaccines. Okay. Now, antibiotics, what are antibiotics? We've heard of this name a lots of times when we fall sick, doctor gives us antibiotics. So what are antibiotics? They are the chemicals which are produced by microorganisms, and these microorganisms stops the growth or destroys the disease-causing microorganisms or microbes. Okay, these chemicals are produced from microorganisms, and the microorganisms which are producing these chemicals, those stops the growth of the disease-causing microorganisms. Okay, these are useful microorganisms which stop the growth of the disease-causing microorganisms. That is, harmful microorganisms growth are stopped by these useful microorganisms. Stop or it destroys also sometimes. Okay. For example, streptomycin is the antibiotic name, which is prepared from Streptomyces griseus. Tyrotrycin, it is prepared from Bacillus brevis. Polymyxin, it is prepared from Penibacillus polymyxia. Okay. Then chloromycetin, it is prepared from Streptomyces venezuela. Then tetracycline, it is prepared from Streptomyces aureofaciens. Now, we have known what are antibiotics and some of the names of the antibiotics. So, to prepare antibiotics, what are the criteria needed for a good antibiotic to be prepared? So, first is it should be able to kill a variety of disease-producing microorganisms. Okay, so the good antibiotic should be able to kill a variety of disease-causing microorganisms. These are called broad spectrum antibiotics, which can kill different types of disease-producing microorganisms. Okay. Second should be that it should not produce undesirable side effects. It should not produce undesirable side effects. Again, it should not kill normal bacteria of the host. That is, the good bacteria which are present in the host, that should not be killed by the antibiotics. Okay, and it should not produce some undesirable side effects to the host. Okay. So these are the three criteria needed for a good antibiotic to be produced. Okay. Then antibiotics are also used, that these antibiotics are, we have now studied that the antibiotics are used to cure certain diseases. Okay. That is, they are against certain diseases, or rather to kill certain disease causing germs, disease or organisms. But there are also some uses of antibiotics, some other uses also there. What are those? If they are used to prepare food, to preserve food like meat, fish, to treat animal feed. Okay, and they are also used to control plant pathogens. Plant pathogens are also killed by antibiotics. Okay, animal feed, treating animal feed is also used by antibiotics. Even to preserve food, antibiotics are used. Clear? So, 4 uses we have found. One is on medicines, that are as medicines for killing or stopping the growth of the disease-producing organisms. Then preserving food, then treating animal feed, and controlling plant pathogens. 4 uses. Now we come to serums. What are serums? Serums are substances having antitoxin of a particular disease-causing microbe, especially the bacteria. They have some antitoxin of a particular disease-causing microbe. Actually, the serum means what? Blood plasma without the fibrinogen, but having numerous chemical substances like antibodies and antitoxins. Okay, without fibrinogen, but having antibodies and antitoxins. That is called serum literally. Actually, it means. But here serum is a substance which contains the antitoxin of a particular disease-causing microbe, especially bacteria. Now, what are antitoxins? Antitoxin is a substance which is produced in an animal body reacting with a poison, that is toxin, which is produced by invading the germs. That means what? When a germ invades our body, it produces a substance, a chemical that is known as poison or toxin. Okay, the germ produces in our body, but this toxin is fought back by certain chemical substance which is released from our body or animal body. Okay, that is known as antitoxin. Clear? Now, what are toxins? Toxins are poisonous proteins that are released by the pathogenic bacteria during their growth or upon their breakdown after they die. Okay, so toxins are what? Those are poisonous proteins which are released by the pathogenic bacteria. That means the bacteria which is invading our or any animal body which is invading. On that, it produces some proteins that is known as which are poisonous in nature. Those are toxins. Okay, when these proteins are produced when they grows inside our body or inside an animal body. When they grow, they will produce, or when they grow or when they break down after they die. When they grow or when they break down after they die, they produce certain poisonous substances, poisonous proteins. Those are known as toxins. Excuse me. Those are known as toxins. Okay. And to fight back our body or any animal body on which the toxin has been produced, the substance which is being prepared by the animal body or human body, that is known as antitoxin. Okay. To prepare the serum, what process is required? To prepare the serum, small doses of bacterial toxins, we take some small doses of the bacterial toxin that is taken and injected into blood of a healthy animal, like a horse. Okay. So small doses of bacterial toxin is injected into the blood of a healthy animal like horse, and that horse's blood, that will produce an antitoxin to neutralize the effect of the toxin. Now the horse's blood is taken out after it produces the antitoxin. This horse's blood is taken out and chilled. Okay. This clear straw-colored liquid separating out is the serum. The straw-colored liquid, the blood is taken out and chilled, and then we will see that there's a clear straw-colored fluid coming out. That is known as serum. That provides immunity to a particular disease. Okay. This serum provides immunity to a particular disease. For example, for snake bite, anti-venom. Snake bite, when the snake bites, it produces a venom. Okay, and to neutralize that, anti-venom is produced from the horse's blood. Okay, then insulin, hormone insulin. Okay, when somebody has diabetes, doctors suggest to take insulin as injections. So this insulin hormone is prepared from E. coli. It's cherry coli. Okay. For hemophilia A, blood clotting factor 8 from bacteria which are genetically modified. Bacteria which is genetically modified, that produces blood clotting factor 8, which is used for treating hemophilia A. For treating hemophilia B, factor 9 is prepared from genetically modified bacteria. Okay. So these are all serum examples. So understood, I think, what is serum, what is antitoxins, toxins, and antibiotics. Okay. Now we come to vaccines. Now, vaccine is a preparation. It's also a chemical. It's a preparation which consists of weakened germs or dead germ substances. Okay, it's not rather chemical. It is the weakened germs or dead germs substance. So it's a preparation which consists of what? Weakened germs or dead germs. Now, to introduce that weakened germs or dead germs into a person's body, into a person or into an animal body, that is so that this body develops a resistance to a particular disease by developing antitoxins. That process is known as vaccination. Okay, for example, cholera, typhoid, measles, chickenpox, etc. These vaccines are used. For typhoid, killed bacteria is used, and the vaccine name is TAB vaccine. For tuberculosis, living weakened bacteria is used, which is known as BCG vaccine. Okay. So this is about vaccine. I think you are clear. Next, we come to toxoids. Okay. Now, inactivated toxins which are inactivated, that has not been activated of a particular bacteria. which stimulate the production of respective antibodies, like producing immunity against diphtheria and tetanus. So, inactivated toxins are known as toxoids. Okay, inactivated toxins of what? Of a particular bacteria, which stimulate the production of respective antibodies. Okay, for example, producing immunity against diphtheria and tetanus. Now, this is about role in medicine. Okay, now we come to role in agriculture. In agriculture, bacteria are used in nitrogen fixing, nitrifying, and also denitrification. So nitrogen fixation, nitrification, and denitrification. Okay, and totally these three processes constitute the nitrogen cycle. Okay, so let us come to the diagram. How all these processes occur. Now, first we come to nitrogen fixation. We know that atmosphere contains nitrogen, free nitrogen. Okay. Now, rhizobium bacteria is there, or you can say soil bacteria, which is present in the soil. Okay, and that remains also, they are present in the nodules of certain leguminous plants. Okay, what do? They absorb, they take the atmospheric nitrogen and they convert that nitrogen into nitrates in the soil. This is done by nitrobacter. Okay. Then azotobacter or clostridium, these are also two bacteria which convert hydrogen, which is present in the air. Hydrogen takes the hydrogen and it combines with ammonia to form amino acid. Okay, it combines hydrogen with ammonia to form amino acid. Okay, and amino acids and nitrates, amino acid and nitrates, and which makes the soil fertile. Okay, so amino acids, and then it converts to nitrates, and then it makes the soil fertile. Okay, so because the soil contains the nitrates, so it will be very useful for the plant growth. So this is done by azotobacter and clostridium. And rhizobium, they absorb nitrogen from the soil, atmosphere and convert it to nitrates. This is known as biological fixation. Okay. Then industrial fixation is also there, where ammonia, which is present in the soil, that is first converted into ammonium compounds, and then they are converted to nitrites in the soil by nitrosomonas bacteria, and then this nitrosomonas bacteria convert nitrites to nitrates by the nitrobacter. Okay, so ammonia of the soil changes to ammonium compounds and then to nitrites by nitrosomonas bacteria. And nitrosomonas bacteria converts to nitrites, and nitrobacter convert nitrites to nitrates. Clear? Now, this nitrification, this whole process from ammonia to nitrates, that is known as nitrification process. Okay, that is ammonia to ammonium compounds, then to nitrites, then to nitrates. This is known as nitrification. Nitrification, generally, in nature, this is an industrial process, industrial fixation, but in general, plants and animals, when they die, okay, when they die, their decayed living matter, the living matter, after the death, the matter, the remainings of the plants and animals, that decay. And then that releases what? Ammonia, which changes into nitrates. That is, nitrification occurs naturally. Okay. Now, after these nitrates are formed, they are used by the soil. But there are certain bacteria which convert these nitrates into free nitrogen. Converts the nitrates to free nitrogen. Okay, that is, for example, Pseudomonas bacteria that converts nitrates to free nitrogen, and that is known as denitrification, the process of denitrification. And this denitrification reduces the fertility of the soil. Okay, so this is nitrogen cycle. These main three processes, that is nitrogen fixation, nitrification, and denitrification. Atmospheric nitrogen coming down to form nitrates and then again returning to atmospheric nitrogen. So this total is nitrogen cycle, and this is totally done by bacteria. Okay, certain notes are there. See, bacteria help in the decay and putrefaction of dead plants and animals and organic organic excreta. So the plants and animals, the animals, when they release their feces and excretions, objects are there. Those are converted, and even the dead and plants and animals are converted and decayed and converted into certain useful substances. Okay, so this decay and putrefaction is done by bacteria. So what is decay? It's complete breakdown of the organic matter without any foul smell. And putrefaction is incomplete breakdown of the organic matter with a foul smell. Okay. Then in the sewage treatment plants, where sewage bacteria converts human excreta into manure for agricultural fields, and the gas which is released, that is used for cooking. Okay. Then the gobar gas or biogas plants, they use fermenting bacteria to degrade the cellulose of the cow dung. The cow dung has some certain cellulose material. So that is degraded by the fermenting bacteria to release an inflammable gas, which is mainly methane, and that is used for cooking. And they supply rich manure also after being treatment. This biogas that helps in this inflammable gas comes out, and also the the remainings they supply as rich manure for the agricultural field. Okay, so that is done by, in the biogas plant, fermenting bacteria are used. Okay, then several bacteria synthesize vitamin B and vitamin K, and even some help in the digestion of the cellulose. Certain bacteria are used for digestion of the cellulose. Okay. Now we come to, this is about agriculture. Now we come to the industrial root. Okay, like tea leaves. Tea leaves, curing of the tea leaves, that is the flavor which we get, the taste of the tea leaf which we get, that is done by fermentation, and that is this fermentation is done by bacteria. So bacteria is used here for curing of the tea leaves to get the flavor and the taste of the tea. Then tanning of leather. Okay, that is the perishable parts of the leather that is decayed. Okay, so that is done by what? Bacteria again. Okay, the skin, the hair, the parts of the skin, that is the hairs. Okay, those are broken down by the bacteria. That is tanning of leather, which is used for, then this leather is used for different other purposes in our household uses also. It is used for making shoes, bags. Okay, then we come to, this is about the all the benefits of bacteria which we get in our day-to-day lives. Now we will get some harmful effects of the bacteria we will discuss now. Okay, three main harmful effects are there. Rather, we can say four harmful effects are there. One is spoilage of food. Second is plant diseases. Fourth is animal diseases. And third is animal diseases. And fourth is bioweapons. Okay. So first we come to spoilage of food. Some saprophytic bacteria, they spoil the food. Okay, like rotting of the vegetables, fruits, pickles, meat, cheese, etc., butter, etc. These are all spoiled foods. So that is done by bacteria. Then ferment cooked food, vegetables, fruits. Then food poisoning is caused by bacteria. For example, botulism. Botulism is it's a spoilage of food. It's a food poisoning which is caused by Clostridium botulinum, and it is in the canned or tinned or sealed foods, no? Those food cause food poisoning. That is done by Clostridium botulinum, and this disease is known as botulism. Okay. Then we come to plant diseases. Plant diseases like black rot of the mustard and cauliflower, that is done by Xanthomonas campestris. Then bacterial blight of cowpea, that is Xanthomonas axonomopodis. Then blight of paddy is Xanthomonas oryzae. Then crown gall of sugar beet rather, by Agrobacterium tumefaciens. Okay. Then wilt of tobacco by Phytobacterium. Then citrus canker, Xanthomonas citri, etc. Okay, these are all diseases which occur in plants by bacteria. Now, animal diseases. Excuse me. Cholera by Vibrio cholerae, typhoid by Salmonella typhi, syphilis by Trypanosoma pallidum, tetanus by Clostridium tetani. These four examples are of human diseases, okay, by bacteria. Then anthrax by Bacillus anthracis, where there is a swelling of the body and milk yield is reduced. Then bovine tuberculosis, that is caused by Mycobacterium bovis, where there is a dry husky cough, and the lungs are affected. Okay, these two are examples of cattle diseases that occurs in cattle by bacteria. Okay. Now we come to the fourth harmful effects, that is the bioweapons. So what are bioweapons? These are microorganisms, especially bacteria, which promote disease or death in people, animal, or plants. Okay, these are microorganisms which increases the disease or death in animals, plants, and people. These bioweapons are also called germ bombs. Some countries, they prepare these germ bombs, and mainly they release certain disease-causing germs like anthrax bacteria, which cause epidemic death generation after generation. Okay, for example, anthrax, smallpox, salmonella, Yersinia pestis. Okay, these are all used as bioweapons. Okay, biological way of killing habitats. Okay, used against humans, plants, animals of a country. Okay. Now we have studied the harmful effects of the bacteria. We have first point of the harmful effect was spoilage of the food. We know food are spoiled if we expose them to bacteria, to that environment where bacteria can grow. To preserve the food so that it does not get spoiled, certain methods are used. So what are those methods? Food preservative methods. First, sterilization. Second, salting. Third, dehydration or drying. Fourth, eradication. Fifth, pasteurization. Sixth, refrigeration. And seventh is strong concentration and chemicals. So first we come to sterilization. Okay. First is sterilization, where the substance is kept in water and it is boiled or heated at a very high temperature. High temperature, what does it do? It kills the bacteria except the spores. How much high will be the temperature? Around 110 degrees centigrade. And if it is more than 110 degrees centigrade and increasing pressure, that is, if it is treated in cooker or autoclave, then the spores even can be destroyed. Okay. Then the canning industry uses this packing and sealing food. Before packing and the sealing food, the canning industries, they use the sterilization method to keep the food away from bacterial growth, from getting spoiled. Next, we come to salting. Salting is another way to preserve food. Here, the food is preserved like fish, pickles, cheese, and so on. For long periods, it can be kept. It can be preserved. Biodegradation is prevented by this method, and this method is also called curing. We covered the substance with extra concentration of salt so that the bacterial growth does not take place. Okay, like that, we can keep it for long periods. And then dehydration or drying. This is, as the name suggests, there is no water. So dehydration means no water, no dehydration. Hydration means with water. Dehydration means without water, or drying means without water. So it should be kept dry. So without water, generally no growth of microbes takes place. And this process preserves grains, meat, fish, vegetables. Like, for example, is dried milk powder. Then irradiation. Irradiation, that means what? Radioactive radiation or ultraviolet radiations are used to preserve the food. Okay. For sterilizing certain foods, it is used. Radioactive radiation is used for sterilizing certain foods, but this method is not much used. Whereas ultraviolet light is used for sterilizing certain schools, hospitals, factories. Okay. So that will kill the mold, spores, and even bacteria. Okay, mold, spores, and they are used in the sterilizing the schools, colleges, hospitals, factories, and even the bakeries also. That can be sterilized by this ultraviolet light. Okay, this is irradiation. Then pasteurization. Pasteurization is the process to sterilize milk. So partial sterilization you can call, usually of the milk. Okay. So in this process, milk is heated to about 60 degrees centigrade. For 30 minutes, it's heated and then suddenly cooled, chilled quickly. Okay. So majority of the bacteria are killed here, but it will not kill total. Excuse me. So it will not sterilize the whole milk. So it contains some of the bacteria which can cause souring of the milk. Okay. So souring bacteria are still left in that milk, which can do harm to the milk. Okay. These bacteria do not multiply in the cool temperature. So the substance is cooled, the milk is cooled, so that the bacteria, these souring bacteria, they will not multiply. Okay. Then we come to refrigeration. Here, bacteria and mold, they do not grow or multiply at below the freezing point of water. That is, freezing point of water is 0 degrees centigrade. So below 0 degrees centigrade, if a substance is refrigerated, then the bacteria and molds, they do not grow or multiply. Okay. So this process can preserve milk, fruits, vegetables, fish, etc. for a long time. Then short-term storage in our domestic users, 0 to 5 degrees centigrade, it can be cooled to. And in long-term storage in industrial large-scale storage when it is required, then it's stored to a freezing temperature of -20 degrees centigrade to -30 degrees centigrade. Okay, another process is there that is strong concentration of chemicals like jams and pickles. Okay, they have strong concentration of sugar and salt respectively. So jams contain strong concentration of sugar and pickles contain strong concentration of salt. Okay, so that can preserve them. Then increased concentration of solutes. If what happens actually in this, when the concentration of the solute increases, that will plasmolize the bacteria or mold and that will kill them. Okay, plasmolize means it will take away the water from the bacteria or the mold from the cell. Okay, and it will dehydrate it. So it will die. Now, sodium benzoate is used as preservative. It's added to tinned foods or squashes. Sodium benzoate is a preservative which is added to tinned foods and squashes to make it germ-free. Okay, so this is the end of the chapter. I mean, not end of the chapter, end of the second part of the chapter, that is bacterial economic importance. Okay? So both the useful and the harmful effects, and how to preserve food we have also discussed. So next part will be on fungi. Okay. So till then, please do go through this video and the previous one also. If you have any doubt, please do write in the comment box, and if you're liking the video, share with your friends so that they can also be benefited from these videos. You can write also at my email ID, that is kasturi74@gmail.com. And if you are a new viewer to my channel and not subscribed yet, please do subscribe, because that will notify you as soon as you as soon as a new video is uploaded. Okay, and please do meditate before you start your day, and do maintain a chart so that will help you to note down how much you have completed in your whole day and your progress in day by day. Okay, and that will help you to complete your syllabus for your examination, and help you in your tracking your studies. Okay, so thank you for joining. Have a good day. Thank you.